The River Rhine is one of Europe's most important rivers, economically, culturally, but in particular ecologically. Originating in the Swiss Alps, it flows through and connects 6 countries before reaching the North Sea at Rotterdam. Over thousands of years, the Rhine has shaped a unique landscape. Our focus is on the Rhine as a natural habitat for plants and animals, with a particular attention to its ecological quality. As settlement along the Rhine Valley progressed, human influence on the river increased. Today, its original state is barely recognizable in many areas. Intensive agricultural use for food and feed production, strong population growth, and urban sprawl dominate the landscape along the river. Additionally, the Rhine has become one of Europe's most critical industrial transport corridors. All these factors have left clear marks on the ecosystem. To assess the current state of the river and its biodiversity, a comprehensive study was conducted in collaboration with the International Commission for the Protection of the Rhine. Scientists collected samples at a total of 32 sites along the river to analyze changes in species composition, from source to mouth. Using measurement instruments, key physicochemical parameters such as pH, oxygen content, conductivity, and water temperature were recorded. These are all crucial factors influencing the living conditions of aquatic organisms. In addition, 2 liters of water were collected at each site and examined for traces of DNA. All organisms, fish, crustaceans, insects, release DNA into their environment through skin cells, excretions, and other biological materials. This is referred to as environmental DNA or eDNA. These genetic traces provide a unique fingerprint for each species. Similar to forensic methods, these traces can be detected in water bodies. For this purpose, the water samples were filtered on site and then analyzed for eDNA in the university laboratory. This method, eDNA metabarcoding, enables the detection of a wide range of species, including many that are otherwise rare or difficult to identify. It allows for a comprehensive snapshot of biodiversity in the river. Our main objectives are to monitor changes in biodiversity along the Rhine and to explore the potential of this innovative monitoring method. Using eDNA metabarcoding, it is possible to sample a large river system, like the Rhine, in just a few days, while obtaining high-resolution data on species presence that can be analyzed within weeks. The findings from this research can provide crucial insights for environmental monitoring, for water management and nature conservation. They can help to understand the spread of invasive species, assess the health of native populations, and support the evaluation of the overall ecological state of the river. The data have arrived. What we see is that we were successful in capturing environmental DNA data along the whole river. We find traces of 26 fish species, and we find traces of a couple of hundred invertebrate species. However, now a detailed analysis of the data needs to be done. The overarching aim for us as environmental researchers is to provide data that are reliable. And what we need is reliable data to see how the River Rhine changes, where is biodiversity changing in space and time, in order to provide these data for practitioners that are in charge of implementing measures for the sustainable development of the River Rhine.